Hey, this is Miss Mannix, and what we're going to look at today are the light-dependent reactions or the light reactions of photosynthesis. And before we start, it's important to remember that photosynthesis occurs in two steps, the first being the light-dependent reactions or the light reactions, and the second being the light-independent reactions or the Calvin cycle. So oftentimes when we're looking at light reactions, it's easy to break them up into kind of two steps by looking at the photosystem. So we're going to start with photosystem 2. And remember, photosystem 2 comes first because of the order that the photosystems were discovered. So what happens is when light gets absorbed by chlorophyll, and it's important to remember that chlorophyll is bound to proteins called antenna proteins, which you can't see here, but they surround the reaction center, which is P680 in photosystem 2. So what's going to happen when that photon of light gets absorbed is it's going to excite electrons. So electrons are going to go up in energy level. They're going to reach their excited state. And what happens is that electron is going to get transferred from chlorophyll into the reaction center. So what's happened so far is we've transferred the energy from the electron into the reaction center. And what's going to happen at the reaction center is the electron is going to get transferred to the primary electron acceptor. So our reaction center has now become P680 plus because it lost an electron, and the electron is at the primary electron acceptor because it's going to help it move on to the electron transport chain. So how do we replenish this, right? Because we lost an electron, we need that electron back in order to keep this system going. So this is where water comes in. So what's going to happen with water is going to be split. And so water is going to be split into hydrogen ions and oxygen, but it's also going to release its electrons. And those electrons are going to be used to replenish the reaction center. So we're going to reduce P680 plus, it's going to become 680 again. And why those hydrogen ions are important is because those hydrogen ions are going to be released into the thylakoid space, that inner area of the thylakoid membrane where light-dependent reactions are taking place. And that's going to be helpful in the next step in order for us to create ATP. So the one oxygen atom that's produced from when water is split is immediately going to go bind with another oxygen atom to create oxygen gas. And then that oxygen gas can be released through the leaves stomata and into the atmosphere. So what's going to happen now is our electrons are going to pass from photosystem 2 to photosystem 1 through the electron transport chain. And that's what we're going to take a look at. So that electron's going to move from the acceptor to the first step of the electron transport chain, which is called plastoquinone, which is a protein. It's then going to transfer to the cytochrome complex, which is an enzyme that just assists in the movement of electrons. And then the final step in this first electron transport chain is plastocyanin, just another protein. So what's happening here is the fall of the electrons from their excited state to their ground state from photosystem 2 to 1 is providing the energy to form ATP. So when we're splitting that water to replenish the electrons in photosystem 2, we're creating a concentration gradient of H pluses inside the thylakoid space. There's also some hydrogen ions outside of that thylakoid membrane, and when that energy is being released from the electrons, we use that energy to pump those hydrogen ions across the membrane, and then we're able to use those hydrogen ions through ATP synthase. So what's going to happen, we know when we have a high concentration of ions on one side of the membrane, it wants to diffuse through, in this case, facilitated diffusion to that low concentration. So when hydrogen ions diffuse through ATP synthase, it allows ADP and phosphate to join together to create ATP. And this ATP is really important because it's going to power our light-independent reactions, or the Calvin cycle, in step 2 of photosynthesis. So far, we've looked at how light is absorbed by photosystem 2. It's going to excite those electrons. The electrons are going to move from the reaction center to the primary electron acceptor and then down the electron transport chain. Those electrons will go into photosystem 1, which we'll talk about on the next slide. To replenish the electrons in photosystem 2, water is split, creating oxygen, hydrogen ions, and as well as electrons. And then we use that energy from the electrons being pushed down that electron transport chain to pump the hydrogen ions in to go through ATP synthase to create ATP. So let's look at what happens in photosystem 1. Similar, we have those electrons coming in from the first electron transport chain, and we also have light being absorbed by our chlorophyll molecules. So right now, the electrons that came in from the photosystem are back at their ground state. So we're going to use that light energy to excite the electrons, which will move into the reaction center, which in this case in photosystem 1 is P700. We're going to lose that electron to the primary electron acceptor, making the reaction center P700+. And then the electrons are going to go down a second electron transport chain. The proteins are a little different in this one. We have ferredoxin, which then moves the electrons into NADP plus reductase, which is going to help us make an electron carrier molecule, NADPH. So NADPH is important because it's going to help move energy from the light-dependent reaction into the light-independent reaction. So like I said, NADP plus reductase is going to catalyze the transfer of electrons from ferredoxin to NADP plus to create NADPH. And so that is what we see here. So what we've done is we have taken light energy and we've put it in the form of chemical energy in NADPH and ATP. And ATP and NADPH are what we use to power the second part of photosynthesis, the Calvin cycle. So just as a reminder, our inputs and outputs, we input water, ADP, could also add plus a phosphate, and NADP plus, and we got out of that oxygen, ATP, and NADPH. So we'll do one summary slide, kind of like the elevator pitch. If someone asks you what happens in light reactions, you're going to say the goal is to convert that light energy or solar energy into chemical energy. And we did that in two forms, NADPH and ATP. We split water. The purpose of splitting water is it provides electrons and hydrogen ions, and it also releases oxygen. Light was absorbed by chlorophyll molecules, which helped those electrons and hydrogen ions from water go to an electron acceptor, NADP+, to make that NADPH. And then we also generated ATP by the phosphorylation of ADP when those hydrogen ions were pumped through ATP synthase from a high to a low concentration gradient. All right, that's all for now.